Hi everyone! Alright, so today what I want to talk about is how do we go about downloading SRAs. SRA stands for Sequence Read Archive, and these are different data sets. Specifically, the one that we will be working with is the RNA-seq data set. So how do we go about finding the SRA to begin with in order for us to even download them? So NCBI has the largest publicly available repository for different sequencing data sets. So go ahead and click on this link right here. So this link takes us to the NCBI page in order for us to access data within this repository. So we're going to enter the GDS accession code that we are interested, which is GSE150377. And here it will take us to a page that shows us different collection of samples. We are going to specifically look at this first link right here, which is going to look at BROC1 cells. I selected this data set based on your interest into looking to BROC1. Just a caveat, I have not worked with this data set prior, so I do not know exactly what we will find in our research of looking at this data set, nor do I know the quality of this data set at this time. Okay, so let's go ahead and open up this page and it will take us to this landing page right here. So here you're given the title of the data set, the organism, the experiment type, so expression profiling by high throughput sequencing. So we're going to be looking at a sequencing data set and a summary of the research project. So again, I selected this data set because of your guys' interest with working with BROC1. So we can see that this is going to be a transgenomic analysis. So using RNA-seq, this study will look at two different types of cell lines, a BROC1 proficient and a BROC1 deficient. And there's going to be treatments involved with this data set as well. So these cells are going to be treated with trizine compound CT1913 as well as its metabolite. The citation listed here below is the paper, the peer-reviewed paper that developed these samples. So go ahead and scroll all the way down and we will see more information about the samples themselves. Notice the bioproject number, the SRA, and down here, the SRA run selector. This is the link that we're going to click on. So before we go into that, I wanted to briefly discuss SRA submission. I wanted to briefly talk to you guys about the anatomy of an SRA submission, just so that we understand what all these different numbers that we're seeing on the next few pages mean. So the bio project, which was that first link that I point you to, that had the PRJN prefix, this refers to the bio project, which will provide us more information about the research project, including the title of the experiment. Next, there was a link that was a biosample with the SRP. SRP is going to be the study. So that will be the study that we will be looking at. The experiment is the SRX, the run is the SRR, and the submission accession is the SRA. The SRA is going to contain information on the library sample, the instrument, and is directly associated with the sequencing data. The samples, the SRS, can be shared actually between different studies, and thus they can be deposited into database by multiple submission. Each experiment, that is the SRX, is unique for the specific sample. So in this illustration, for example, we can see that there are different sequencing library that were prepared, right? So this one versus the one over here. And each one of these sequencing library has a different type of biological sample, may or may not have biological replicates, may be part of a treatment group, for example, treated versus non-treated, can have different type of sequencing strategy, for example, ChIP-seq versus RNA-seq, may have been sequenced on a different type of instrument, and the samples themselves, the reads, may be paired end or single end, that is unidirectional sequencing. Go back to the previous page, now that we've looked at what the anatomy of an SRA submission consists of. Back on this page, I'm going to click on the SRA run selector because I'm interested in gathering the raw sequencing data. So here is information about this particular bio project. Interested in gaining more information about the research description itself, you can click onto this link. But let's look at that first box, the common field. So again, this is the RNA-seq data set that we're going to be looking at. We're interested in the FASTQ, but this is the type of file type that are stored for this data set. We can see that it was sequenced on the NextSeq instrument. The libraries are paired end. This is the biological sample, is from Homo sapien. And this is the associated SRA study. Scrolling down, we get to this box right here, which is select. So from here, we can download the metadata and the accession list. We won't really be using this accession list, but let me just show you what it contains. So here, these are the different SRR numbers, accession number that are associated with this study. If we scroll down, we can see those same accession number right here. So here are the SRR run study. Mentioned in the previous slide, each sample SRX experiment has an associated SRR run as well as the SRA. When we download this later on, we will see that each SRA is going to have two sets of reads associated with it, _1 and _2. One of these will be the forward and one will be the reverse strand. Now, there are two ways in which you can download these SRAs. First option, which is the one I'm going to go over in this video, is to use the SRA Explorer to get the FTP URL link and then to go into the terminal and use curl to fetch your SRA data. The second way is to use the NCBI SRA toolkit. I can go over that if you guys are interested, and I will do that in a future video. So with the first option, we can use the SRA Explorer using this link right here to fetch the FTP URL and then enter that FTP URL on the terminal in order for us to be able to download the data set. Coming back to this page, I'm going to go over how to go about doing this for one data sample. So we're interested in saying this first one right here. So go ahead and copy that and open up that link that I provide you for the SRA Explorer. And we're going to go ahead and enter in that SRR run that we're interested in. So let me search. If you scroll down, this will give you information about the experiment title and the accession number right here, information on the read, and it will also tell you how many bases are included in this data set. So go ahead and click that and add it to your collection and open up the data set right here. And we're going to look at the bash script right here. So this bash script is giving us information on the FTP that we need in order to download. So I am just going to copy this first line. I will talk to you further about what is included here, but let's just copy this first line right here. Go ahead and open your terminal. So I'm going to type in terminal. and this will take you to this terminal. If you need a refresher on how to write command lines, go ahead and watch the previous video on Unix command line, but I'm just going to dive right into it. So this will take me to the home directory, and I actually want to download this into a different folder. So I'm going to go ahead and make a directory. Call this summer MBT. I'm going to go ahead and go into that folder right there. And let's go ahead and make another directory that I'm going to call raw. So I recommend that you organize your directory in a way that you can keep the original file separate from the files that you will be working with. This is not as important here because we can always re-download the file again if needed. But imagine if these were your raw data that you got from the sequencer. You want to make sure that they are listed in a directory folder that is separate from the files that you'll be working with, because you do not want to risk the chance of these files being overwritten or deleted. So for good practice measure, I'm going to place these files that we're downloading into a raw file. So let's go ahead and cd into this raw file directory. Okay, so if I look, this is the directory that we are in right now, which is the summer MBT and raw directory. Let's go ahead and paste that line in, and I go over what this command is going to do and what these arguments mean. So we're going to use the curl command line in order to be able to download. So if you want to find more information about curl itself, you can use this link right here. And I found this cheat sheet on this GitHub right here. So we're using two arguments, the argument -L, which is going to follow the redirect, so follow the FTP link that's provided, and then -O for output. So this is going to be the output file name that we will want to place into our raw directory. Okay, so let's go ahead and run that. And you can see here that is providing information for you on the run itself. So how much time does it have? What is it downloading? And so on. So we'll come back to this in a little bit. But let's look again at that bash script that I showed you earlier. So we had just copied this first line right here. But you can see here that it has a lot more lines. There's 3 lines total for this one script. If you wanted to get all the SRR within that data set, you can actually go to the SRP. I'm going to copy this right here and take it back to the Explorer and paste it in here. You can see here we have all the 12 samples. So I add this to the collection and I go here, I can get the FTP URL for all the samples. Notice that there are more than 12 samples here. This is because, as I mentioned earlier, this is a parent library. So for every sample, there is an _1 and an _2. So one of these data set will be the forward strand and the other one will be the reverse strand. So here is the shell script that we can use to download all 12 samples. I am just going to go ahead and copy this. I'm going to go ahead and open up another terminal window. And I'm going to go into that folder that we had created. I'm going to go ahead and make another directory, scripts. And I'm going to create a new shell script called SRP 261276. So this is the SRP that's associated, and I'm just going to call this SRP download. And because it's a shell script, I'm going to end it with the suffix .sh. And we're going to go ahead and just copy that script in here. I'm going to just Control V. And let's go up to the top. So you can see each one of these command line is a curl command to download the raw data file. And this first file, I'm going to press I for me to be able to edit this. So I'm going to change this first line real quick to a shebang that my computer recognized. So this first line is called a shebang, and it basically allows the computer to know that this is a bash script. Following this line is my actual command line. So this is what I would put on the command line, but there's multiple command line. So when I run the script, it's going to recognize there's a shell script, and it's going to start going through line by line to retrieve the command line itself. So now that I am done with copying, I'm going to press Escape, WQ. This allows me to exit and saves the file. Okay, so if I want to run this shell script, all I have to do is press SH and the file, and it will run the file. At this time, the way that I have the shell script written though, it's going to download each one of these files into this script folder. So if I want to have it run it and point the download file to the other directory, my raw directory, I'm going to have to add a little bit more to the shell script, which I'm not going to do at this time. If you are interested in that, you can let me know, and I can show you how to go about that. But just be aware that because I'll be running this shell script in the script directory, this is where the files will be downloaded to. You can download it to the script file and then afterward move it from the script folder into your raw directory folder, or you can add an additional line that tells the shell script where your raw folder is on your local machine, and then tell it to download the output file to that folder itself. But the focus right now is to kind of show you how to go about downloading the SRA file in general, so I won't go too much into shell scripting at this point. So let's come back to this window right here. We can see that our file has downloaded 100%. So if we go ahead and type in ls, which is for list, we can see the file is listed right here. Actually, I'm going to do, this just provides me more information. So this is the file that we just downloaded, and it tells you how big the file is, as well as who the owner is and permission, which we can go into further in Unix. But what I want you to recognize is that this right here is just the _1 file. So in order to move further with regards to analysis, we need to have both the forward strand and the reverse strand. So we're going to go ahead and repeat this for the complementary pair file. So this is _2. Perfect. So I'll go ahead and just let it run like this in the background. But we will need both of these files for our future analysis. So go ahead and download one of the samples, but make sure you capture both the _1 and the _2. Okay, so we just finished option 1 of how to go about downloading the SRAs. In the next video, we'll go over option 2, which is using the NCBI SRA toolkit in order to download the SRA. Personally, I recommend that you go ahead and use this option 1, which is looking at the SRA Explorer in the command line. And the reason why I say that is with option 2, when you're using the SRA toolkit, it requires you to download the application and get it set up on your computer. That can bring with it some headaches that I will go over next time on how to go about overcoming some of the challenges that you may have trying to set up the SRA toolkit. For now, I would recommend just using the SRA Explorer and command line, but it is good for you to look at the other video that shows how to go about setting up the toolkit, just because one of the things that you have to do in bioinformatics is learning how to deal with packages and library and setting that up. And there's always going to be some sort of challenge that you might have to face. So it's kind of good to know how to go about dealing with some of those challenges. Okay, so that's it for option 1. And I hope that this video was helpful and clear. And I will see you guys next time. All right, thank you. Bye.